Hello, BNG319 class. This is the final lecture here for the final exam review. If my computer ever gets up and going here. The old thing is having a tough time these days, but it'll make it. There it is. Alright, so we left off, actually we hadn't finished our talk on the chemical conjugation. Just want to finish that up. Starting at the when we talked about click chemistry, and an example of this is this copper catalyzed azide alkyne cycloaddition here. And you can see you have an azide molecule and an alkyne, and copper acts as the catalyst, and it makes this triazole molecule. And these green and blue ovals represent any kind of chemical part, functional group, whatever, that you want to bring together in one molecule. And the click chemistry reaction puts them together in a single compound. And this is a unique reaction in that the azides and the alkynes are going to react with each other to make this triazole, and they won't react with anything else. So you can use this a lot, and of course we remember from our drug discovery, drug design flipped class, we made some hydrozones. This could be used in a similar situation, making a bunch of compound libraries and mixing and matching them and finding active compounds in that way. So this is a copper catalyzed azide alkyne cycloaddition. Neither of those things, neither azides nor alkynes, are found in nature. It has what we call completely orthogonal reactivity, in that neither azides nor alkynes react with anything else. They don't react with other functional groups. And it's because of drug discovery, drug design, and other types of things. The reaction has found widespread use in being able to add pieces to existing compounds here, using this click chemistry type reaction. The disadvantage of this is that we can't have cells do this. Most cells are going to be killed by the amount of copper, the toxic amount of copper that this would need. And so this is chiefly done out of cells, ex vivo, or in situ, or whatever we want to call it. It's ex vivo. So we can't do this in cells. And just one last thought on click chemistry. You will see a click chemistry problem on the final, so learn it. One application for chemical conjugation is known as ELISA, enzyme-linked immunosorbent assay, ELISA, and it's used for a wide variety of things. It's especially used in the lab to find molecules that will react or conjugate with other molecules. So basically this relies on a molecule with an antibody domain and a molecule with an enzyme domain. So you have a certain antigen on a surface of a well of a 96 or 198 well plate or whatever, and you put an antibody on it. And if an antibody will interact with the antigen, it'll attach to the antigen. Then this other antibody comes along and attaches to this antibody. So we know there's a reaction going on because this red antibody here has an enzyme. And we can use the enzyme, we can add some substrate, and the enzyme will turn this whole solution a different color. Then we can see, hey, this well is an entirely different color. Therefore, the interactions must be taking place because we have the antigen that's attached to the surface of the bottom. Our patient antibodies have attached to that antigen, and our added enzyme labeled antibody has attached to the patient antibodies. And there's a variety of ways we can do this, and I do I shouldn't say this, but I do a bit of ELISA in 312. I assume whoever teaches BNG312 next semester will continue that. Hopefully they do. Another thing is cisplatin. It's one of the oldest cancer drugs, and you can kind of see how it works here. This is conjugating to DNA. You have cisplatin that conjugates to DNA, and it'll conjugate to 2 nitrogen bases in the DNA. You can see the chlorides here. And they will basically tag DNA. And what this can do, and why it's been used as anti-cancer drugs, this conjugates with DNA, can inhibit DNA replication, which is kind of a good idea in cancer cells. It can inhibit transcription, cause cell cycle arrest, and even result in cell death. And if you're talking cancer cells, this isn't too bad an idea. And where this cisplatin attaches, this guanine in the N7 position on the guanine nitrogen base in DNA. So it'll attach to those specific positions on the nitrogen base, and it will potentially do all these things. And of course you can imagine that this is not specific for cancerous DNA or cancer cell DNA. So this has to be administered. This, as a cancer drug, would have to be administered very carefully. And again, the antibodies are a pretty good lesson for chemical conjugation. There are a variety of things that can happen with antibodies. It can be conjugated to antibodies. And again, ELISA is born out of this whole chemical conjugation to antibodies thing. We also have used them in the past. We've labeled them with gold. We've labeled them with other types of molecules, some fluorescent molecules, to allow antibodies to bind to whatever antigen they're supposed to bind to, and then they can localize that antigen, again, whatever it is. You know, we can look at it under the microscope in a cell, in a bacterial cell, in a variety, in a tissue, in a variety of ways, in a variety of places here. So these types of conjugates can help us localize things in cells. And you can see they do a variety of things, and you can have cleavable linkers, acid labile linkers, things that can either indicate where something is. You can potentially have cytotoxic drugs involved too, to turn antibody-drug conjugates into therapies. So there's a variety of things that can be done. And if we're talking therapies, we're talking cancer therapies here, the monoclonal antibody will attach to the antigen, which is a cell surface receptor potentially. It will get internalized by the cell. The cell will start to break down the antibody, and it'll release that drug, which is shown here by a star. The star will end up going, the drug will affect whatever it's going to affect, which will then lead to cell death. Okay, so one of the last applications I want to talk about here is chemical conjugation in hairstyling. And this is interesting because we're looking at the protein keratin. Keratin is a key structural protein for hair, as many of you know. And so the general reaction looks kind of like this. We have keratin that is disulfide bonded to itself, or to other molecules of keratin. We add this sort of conditioner, and what this does is it removes the disulfide bonds. It reduces keratin here. So we have 2 keratin molecules that have sulfhydryls, free sulfhydryls happening, and then we style our hair, and that's kind of the basis of what we call perms or permanents. And then we add hydrogen peroxide, which again will help oxidize and reform those disulfide bonds with keratin, like this. And so what does that look like? Here is a drawing of the whole process here. And so what we're adding is ammonium thioglycolate. and that is known as a perm salt. And if you've ever had a perm, or you've ever known someone who's had a perm, or you've been in the presence of someone having a perm, they kind of smell. They have a sulfury smell. That's because of this ammonium thioglycolate here. And what happens is, again, we have straight hair. We're removing the disulfide bonds in favor of free sulfhydryls. We have these free sulfhydryls in the hair. We wrap the hair, in curlers and such, and then we add the hydrogen peroxide to reform those disulfide bonds, and then suddenly we no longer have the straight hair. We have the disulfide bonds made in curly hair. And so that's theoretically how it is supposed to work. And again, this is ammonium thioglycolate involved in hairstyling. Okay, so quick summary. We talked about mass spectrometry way back when, seems like forever ago now, and the identification of unknown compounds. As you know, mass spec is not going to be in the final. We did talk about chemical conjugation and cross-linking. Some of that stuff will be on the final. It's fairly non-specific. We talk about conjugating proteins to other proteins in terms of things like formaldehyde cross-linking and things like that, and the other cross-linkers we took a look at. They will all react with amines or thiols, and there are a few of those in most proteins. There are some site-specific ones, and we can achieve that by doing this click chemistry. And there are many applications for conjugation like these. Okay, and now to finish our discussion on the topics to know, to study for the final exam. And so I gave you a brief primer on metabolism when last we talked. And so basically it's this way. You are looking at, here are these red arrows going all throughout here. This is the TCA cycle. They're going all throughout, in a circle here. And where we have the red arrows represent, and of course the names are labeled here, which is good. These red arrows represent a enzymatic reaction. And this one happens to be citrate synthase. And what's involved is oxaloacetate plus acetyl-CoA plus water yields citrate, and then you get CoA as well. So you get citrate, which is the product of this reaction, citrate synthase, or the reaction number 1, but then it becomes the substrate for reaction number 2, or for the aconitase reaction, which is citrate is turned to isocitrate. And this is the basis of metabolic pathways, is that they are, they have substrates and then products, and the one product then becomes a substrate for the next enzyme, and then one product from the next enzyme becomes a substrate for the next enzyme, etc., 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 and you go around, and then you have things like coenzymes and cofactors that are involved to make this whole pathway run. And this all runs in a living organism. It runs in a cell, whether that be our cells, yeast cells, bacteria cells, things like that. And so, this pathway wouldn't function, this TCA cycle wouldn't function without things like acetyl-CoA, NAD, FAD, water, certainly, or any of the other carbon, like the oxaloacetate or the citrate, as well. We wouldn't be able to do this. So everything plays its own role. Everything is important, but it's another, instead of showing reactions in the way we have gotten used to seeing them in organic chemistry, we are showing them in more of a flowchart kind of way, I'll say. Hopefully that makes sense. But we've looked at reactions where we have substrate plus substrate yields product plus product. This is similar, except for the fact that we have to really be able to interpret what these lines are. And the more information we're given, the better. So, in here we're given, NAD is converted to NADH plus H plus. We're given that carbon dioxide is liberated as one of the products. So we're given all these things. And because we're given all these things, we have a better idea of what's needed in this pathway. Okay, and of course, a lot of you asked about dehalogenation of PCBs, polychlorinated biphenyls. So this sort of thing occurs anaerobically, and there's a variety of organisms that can do this, and they do this sort of thing for different reasons, mainly so that a microbial consortium can convert PCBs into something edible. So what they do but they have to first dehalogenate the things. And depending on the organism, you can have ortho, meta, or para-chlorine groups removed. And so we are looking at, from a specific point, which we'll call carbon 1 of the aromatic ring. We go from carbon 1, we go to the left. This is for next carbon is ortho. The carbon after that, meta. And the carbon after that is para. And the para carbon is usually directly across the molecule like this from our reference point. And in this case, the reference point is the OH. But then we go back here, meta, because it's 2 carbons away, and ortho is right next door on either side of the, again, our reference point, in this case the OH. And so usually we are looking at reference points what we call carbon 1 or carbon 1 prime. And how do we know what carbon 1 is? Well, carbon 1 is attached to the carbon 1 prime of the other phenyl molecule. And so these are the carbons bonding the phenyl groups together. And so this would be ortho to carbon 1. This would be meta to carbon 1. See this meta, and this is the ortho carbon. There's nothing on it. And then here's our reference carbon. This is carbon 1. This was ortho, meta. Now this chlorine is para to carbon 1. And so when we talk about things that are ortho, meta, para, what does that mean? Well, certain organisms or certain groups of organisms will remove chlorines depending on whether they're ortho, meta, para, or flanked. And so what is flanked? What do we mean by flanked? We talk about flanked, it means they are attached. This is a carbon, has a chloride on it. The carbon that's flanking it, the carbon right next to it, has a hydroxide. So this chlorine is flanked by a hydroxide. So a carbon in a phenyl ring may have a chlorine attached. If it's got something on the neighboring carbon, it's flanked. This hydroxide is double flanked. It's got things on both sides. These carbons, the ortho and the para carbon, this is the meta carbon, these are both chlorinated. They're both halogenated. And so this hydroxide is double flanked because it's surrounded on both sides by different things. And different dehalogenating organisms will possess specific enzyme systems that can remove double flanked, single flanked, or unflanked chlorides. And this is all dependent on the active site of whatever enzymes are used to do this. But for now, for you, it's just enough to know what it means to be double flanked versus flanked versus unflanked. And so let's take a look at that example that we looked at way back when. So if we can remove from this PCB a double flanked meta-chlorine, there's only one of those. This is meta. This is also meta, but only one of them is double flanked. This one has a chlorine on this carbon and this carbon, the 2 neighboring carbons, so it's double flanked. So only this one is going to get removed, and it does. Or if we're looking for a double flanked para, now we have ortho, meta, para. The only para one here actually happens to be double flanked. And so we can remove that. Flanked para, do we have one of those? Yes, we have a para here. It is flanked by this chlorine here. flanked meta. Do we have one of those? Yeah, ortho meta. We have a flanked meta here. Both lead us to the same place. This biphenyl that has 2 chlorines on it in the same place. So we can get there a variety of ways, depending on whether double flanked meta or double flanked para chlorines can be removed, or flanked para and flanked meta chlorines can be removed. And that supposedly goes to aerobic degradation, to be broken up into, and the ring opened, to be broken up into central carbon metabolism intermediates. Okay, a lot of you had concerns about pharmacophores. A lot of you didn't really understand what they were. And a pharmacophore is kind of a fingerprint of a drug or fingerprint of a molecule. And it tells us about the molecular interactions that the molecule performs with the binding site of an enzyme, with the receptor, things like that. Basically, when we do pharmacophore modeling, we're looking for the distance between all the active the active parts of the molecule from each other, and what do they contain, how far away are they from each other, things like that. And this sort of thing, which is all done by computer, can help us figure out what drugs are going to be successful in binding to a particular drug target and is, say, inhibiting or activating whatever it's supposed to do. But what is a pharmacophore exactly? I'll give you the IUPAC definition of that, the International Union of Pure and Applied Chemistry. This is IUPAC. Their definition is an ensemble of steric and electronic features that is necessary to ensure the optimal supramolecular interactions with a specific biological target receptor and to trigger or block its biological response. So that's a bit wordy. Instead, I say, let's just define pharmacophore, because this is not a definition you need to memorize. Let's just define pharmacophore by asking a question. What parts of the drug interact with the target to deliver the response we want? So if we have a drug like this, or if we have a drug that's supposed to have these pharmacophore features here, this has 2, whatever chemical feature 1 is, this distance away, and a chemical feature 2 down here. And we have a drug like this, where we get this molecule, which has 2 different distinct features. We have feature 1, which is apparently a carbonyl group. We have 2 of those, and then we have an alcohol or a hydroxide. Down here is the chemical feature part 2. And these interact, and you can see how they might interact with a binding site, either in a receptor or an enzyme, to deliver some kind of response. These can interact perhaps with an enzyme and inhibit it or something like that. And so let's say we are looking for drugs that behave like ibuprofen, and this is ibuprofen. This is what ibuprofen looks like right here. What pharmacophores could we take into account? There is aromatic group, we can see that. Hydrogen bond donors, there's at least one of those. And hydrophobic interactors, there's many of those. And keep in mind, we need to know whether these groups interact with the enzyme. So there's an aromatic group, as we said. There's a hydrogen bond donor. Hydrophobic, there's some hydrophobic parts to this. That doesn't mean any of these are pharmacophores. Probably at least one of them is, but we have to know more. Here's ketoprofen. So we could compare ibuprofen. There are a whole lot of things that look like ibuprofen and act like ibuprofen. Ketoprofen is one of them. And ketoprofen seems to have a lot of the same stuff. So based on looking at ibuprofen and at least ketoprofen, we can suggest that all of these parts could potentially be pharmacophores. We can't rule anything out yet. So what does that mean for us? Like if I say, look for the pharmacophore, circle the pharmacophore on the exam, the final exam. Well, you have to know how it interacts with the enzyme, the receptor, whatever, how it does its thing as a drug. Also, you could potentially look at a variety of similar drugs that seem to have similar structures and similar modes of action, and make some sort of guess as that. But that's what it would be. It would be a guess. It is not. This is with us doing it on an exam. It's not an exact science. You usually do this by computer to get pretty good models, which are computerized guesses in this case. But anyway, you need to know what is going on. You need to know, and we looked over some examples in class. We looked at AZT. We looked at penicillin. Probably Pharmacophore would be restricted to those things. And you know all about the Pharmacophores there because we talked about them in class. Otherwise, I will have to give you a lot of background, and that's a lot of stuff. Okay, so there was some confusion on the different types of drug designs, receptor-based versus ligand-based. So these are two directions we can go when we're designing a drug based on a target or something else. Receptor-based is indeed based on we know our target. We want to know what binds to it. What binds to it and can either activate or interact in some way or repress. Something like that. So that receptor-based, we will look at it from the receptor. We'll just take in a bunch of molecules and throwing them at this receptor and see what does what we want it to do. What binds to the receptor. Then there's ligand-based drug design. We know the compound that binds to our drug target. Maybe we're fuzzy on the structure of the target and how the ligand binds, but we have a target, we have a compound that binds. And so we can look for similar looking compounds and try to get our lead compound from there. That either will, say, repress an enzyme, that will activate an enzyme, that will activate a receptor, that will act as an antagonist or an agonist, these sorts of things. So it's basically, what do I look at when I want to design my drug? And it depends, again, like pharmacophores, it depends on the information you're given. If you're given a binding site, then try to fit something into that binding site. If you are given a compound and saying, this compound binds to an enzyme that we want to target, look at a kind of a list of other compounds. What looks structurally similar? What has hydrophobic things in the same place as this ligand that we're looking at? What has hydrogen bond donors in the same place? What has aromatic groups in the same place? That sort of thing. So you can ask these questions and come up with a pretty educated guess based on a molecule that looks like the ligand, that looks like the ligand molecule. So receptor-based drug design is essentially given the protein structure and or its binding site, find a new molecule that changes the protein's activity. So what does the binding site look like? Can we stick something in there that's going to interact with key amino acid residues? And again, to do something like this is, it takes a lot of information, from giving you a small binding site, find something that fits in there the best. Usually there'll be one or two things that fit fairly well, that interact with key amino acid side chains, and I'll give you the amino acid side chains. And it certainly won't be as complicated as this thing, but you get the picture. Ligand-based, again, you're given a ligand, find a molecule that changes the protein's activity. So we can look at thymidine, which can be incorporated into reverse transcriptase. Well, what can affect reverse transcriptase of retroviruses? How about something like AZT, which has not an OH, but it has this azide group. And then, the AZT molecule comes in, and, we can stop DNA synthesis by the retrovirus in that way. And if we modeled this off of this, then we've done ligand-based drug design. Alright, surface chemistry is another one. What makes a surface special? Who cares about surfaces? Surfaces are funny things. They have unbalanced interactions. We call there's the surface, and then there's the bulk. So every surface, underneath every surface, is bulk. And the bulk have interactions balanced by molecules of the same type, or the same types. If we're looking at a granite countertop, whatever molecules make up granite. In the bulk of it, in the inside of that countertop, however thin or thick it is, everything is balanced by interactions on all sides, top, bottom, and on all other sides. On the surface, of course, is the surface. It's unbalanced. You've got molecules of the same type on most sides, but not on the top, on the surface. And so these surfaces have residual attractive forces, and they will attract certain things. And you can think, if a surface is, say, plastic, that's very hydrophobic, so you might have residual attractive forces like hydrophobic interactions, or things like that. Or maybe a surface is slightly charged. You have slightly charged surface that will try to go for ionic attractions, salt bridge type of attractions on the surface, because the surface is free to interact with whatever's floating around in the air and lands on it on the surface, whatever is spilled onto the surface, things like that. And so adsorption arises due to the fact that these surface particles of whatever your adsorbent molecule is, whether it's activated carbon or something else, these surface particles are not the same as the particles inside the bulk. And the surface particles are free to interact. The surfaces are unique in that way. The interior of a substance has molecular interactions going on all around it. Bonding requirements are satisfied. The surface of a material, atoms are not wholly surrounded, and they are free to bind to whatever, either by van der Waals forces, electrostatic interactions, hydrogen bonding, things like that. Even covalent bonding in certain cases. And adsorption is usually represented by isotherms, which is, if we're talking about in gas or even another solute, an aqueous solute, the theoretical representation of the amount of solute that's adsorbed. And for gases, it's a function of the equilibrium gas pressure. In liquids, it's a function of concentration. And isotherms tell us how the adsorbent and adsorbate, the adsorbate is the solute, interact under given conditions. Constant temperature, varying pressure, or concentration, depending on whether you're talking about gas or an aqueous solute. And this is represented by the Freundlich equation, which is the relationship between quantity of gas adsorbed per mass of adsorbent and pressure at a given temperature, and is expressed like you see here. It's a very simple equation. You have X over M is mass adsorbed divided by the mass of adsorbent equals a constant, K, times P, which is our pressure, to the 1 over N. N is another constant. And there's an empirical relationship between the amount adsorbed at a given particular adsorbent and pressure at a given temperature. And we can look at curves that have taken place, that have been, of data from different temperatures, and see how these things change. So at constant pressure, adsorption decreases with increasing temperature. And even at a constant temperature, whatever the temperature, saturation is approached, and that's what gives you the shape of these curves. Saturation is approached at high pressure. It doesn't matter what pressure. But Freundlich is not valid for every adsorption situation. There are a variety of isotherms. In fact, surface chemistry could well be a whole course for chemical engineers or even for bioengineers. We can validate Freundlich isotherms by plotting a log-log plot. So the log of our x over m, our mass of adsorbent over adsorbate, versus a log of pressure. You can do this in the aqueous system as well. Log of x over m versus log concentration. And the straight line tells you, if you get a straight line, it tells you that the Freundlich isotherm is valid. Our intercept, our y-intercept, is the log of k, so you can get that constant k from the y-intercept, and the slope of this line is 1 over n. So you get both of your Freundlich constants in this way. Adsorption can be influenced by the charge of the surface, certainly, and also the charge of the solute, or the adsorbate. And so if you have an adsorbate like a protein that has a variety of different charges on the surface, either it's uncharged or it's in some places positively charged in other places, these charges can interact. If you have a negatively charged biomaterial, the positively charged parts of the protein are more likely to interact with the negatively charged biomaterial. And then in that case, electrostatic coupling would be the dominant force. If you have an uncharged portion of a protein and you have an uncharged biomaterial, then perhaps London dispersion forces, van der Waals forces, will interact with the uncharged part of the protein and the biomaterial. Alright, last but not least, we talk about NMR. We just did a homework on that. Everyone did pretty well, at least the ones that I got. So we look at the frequency at which nuclei adsorb, and that depends on the absorb. I'm talking about adsorption. Nuclei absorb depends on the local chemical environment. And so what are their neighbors? We look at hydrogens. This is hydrogen NMR. And so we look at what are the neighbors of these hydrogens. And that tells us where these peaks are going to be. And we didn't really get into that. But it does tell you, knowing the neighbors tells us something about the multiplicity of peaks, which we did talk about as well. And so what does it mean? You're identifying chemically distinct hydrogen atoms. Again, we've only looked at hydrogen NMR. We haven't looked at C13 NMR. Hydrogen NMR is the only one we've looked at. And so the height of a signal is proportion to the number of identical hydrogens. If we look on one carbon, a carbon can bonds to 4 things. In this case, it's bond to another carbon and 3 hydrogens. And so 3 hydrogens here, and we have this carbon bonded to 2 hydrogens and a fluorine. So we'd have 2 distinct peaks for this molecule, and we would have a ratio in terms of heights. The height of the signal would be in a 3:2 ratio. Here we have a carbon that has 1 hydrogen on it, and it's bonded to 3 other carbons. And these carbons on the edges, on the ends here, would all be CH3s because they're bonded to 3 other hydrogens plus this carbon. And so these are all CH3s. There'd be 2 distinct hydrogen peaks in a 9:1 ratio because it's 3 hydrogens, 3 hydrogens, 3 hydrogens. 9:1. This carbon has 1. So they'd be in a 9:1 ratio. Peaks can be integrated. You see those green lines. Those are integration of the peaks. And the signal strength is proportional to the number of protons with that chemical environment, like we said. And so you see some of these here. We have this middle one. We have CH3s and a CH2. The CH3s are presumably this peak. CH2 is presumably this. And the difference in heights is like a 6:2 ratio because we've got three hydrogens, that's 6. And we have 2 hydrogens here, and that's 2. 6:2. And so here, 6:4, we're looking at the 6 of these 2 CH3s. Again, they're in the same chemical environment, so they're one peak. And these 4 hydrogens are basically in the same chemical environment, so they represent a different peak. And so we have 4 hydrogens here, 6 here. 6:2, like I talked about. And these are even. We have 1 CH3 here and 1 CH3 here. We've got an oxygen in the way, so that's keeping these from really influencing each other that much. And so these are in different chemical environments. So you get 2 peaks here, even, because 3 and 3. 3 hydrogens here, 3 hydrogens here. Spin coupling, again, that's the multiplicity of peaks. Hydrogens attached to adjacent carbons affect the possible energy states. So if we have, in this case, we only have 1 peak. All these hydrogens are in the same chemical environment, 1 peak. We've got hydrogens here in different chemical environments now. We've got a CH3 right here and a CH plus 2 chlorides here. And so this is split up into 4 peaks because we have CH3 here. This is split up into 2 because we only have CH. And what does that mean? Signal from a given hydrogen is split by the hydrogens on the adjacent carbon. So for n number of hydrogens on an adjacent carbon, the signal is split to n plus 1 peaks. So let's look at this molecule here again. This one, there are 2 hydrogens on the neighboring carbon. 2 plus 1 is 3. This would be a triplet peak. On this carbon, 2 hydrogens here, 3 hydrogens on the neighboring carbon. 3 plus 1 is 4. That would be a quartet. And so this is what it would look like here. You're looking at the different hydrogens here. So you've got no coupled hydrogens in the adjacent carbon, singlet. You have 1, you have a doublet. You have 2, you have a triplet. You have 3, a quartet. How do you determine what peaks represent what moieties? So you do a couple of things. You look at the peak height, that gives you an idea of what the number of hydrogens are, and the peak multiplicity, the number of hydrogens on a neighboring carbon. And this is probably not the best example because even with what you see here, you don't know that this is a chloride, but you know it's not a hydrogen anyway. So you could be given a little more information to let you know that this is a chlorine, but you don't have to worry about that for the final. We're basically dealing in hydrogens and carbons here. Important note, there was confusion on one of our last quizzes determining the number of peaks. You could determine the number of peaks either way. This was true for the homework. Some of you did do it one way versus the other. One is just you're looking at the number of peaks in terms of chemical environment. If you're just CH2 is a peak, CH is a peak, CH3 is a peak. If you're splitting them up that way, that is correct. I marked that correct. If you're splitting them in terms of what's on the neighboring carbons, or is what's on the neighboring carbon different, I marked that correct also. Pardon me. So I marked either one correct, although I don't think that confusion will happen during the final. But if it does, you can always ask me a question about it. And I'll do my best to help you without giving away the answer. And there's only maybe one, maybe 2 questions on NMR. Anyway, So do it the easier way, just because it's easier, but do it either way and be marked correct, as long as you've got the answer correct in that way, in that particular way. Okay, so that is all I have. I have graded a lot of stuff. I will try to get that all back to you, or at least have that available for you to pick up at my office over the course of the week. And I will let everyone know about that after I post this recording. So good luck studying. If you have questions, please feel free to come see me, email me, what have you, and I'll do my best to help you. The final is again next, not this coming week, but the next week, Monday 3 to 6, and I will see you there.